Bismillahirrahmanirrahim. Today we are going to discuss the major salivary glands, which are exocrine glands. Definitely, they include the list of paired parotid gland, submandibular, and sublingual, as you can see in the picture. Their major production is the saliva, and how much amount being produced daily is 1 liter per day. Its normal pH is 6.5 to 6.9. And the interesting fact about the contents of the saliva, let me tell you, it has organic contents. Let me mention the proteins, amylase, mucin, lysozymes are there, lactoferritin is there, ABO group substances, and rather antibodies IgG, IgA, especially IgM are also there. Then glucose, urea, uric acid, creatine and cholesterol and cyclic AMP are a part of your saliva. And if I have to mention the inorganic contents like electrolytes and bicarbonates ion, let me tell you the sodium, potassium, thiocyanate, calcium, phosphate, chloride and fluoride are there. So the last four which I mentioned are interestingly important for the development of your teeth, right? And this is how they take part in the protection of your oral hygiene. Now we have to learn individually the details of these glands. So let's start with the parotid gland, where it is located. As you can see, these are paired one and located anterior inferior to your ear. And the submandibular gland, which is also called as submaxillary one, is located inferior to the mandible in the floor of your mouth, right? And the smallest among them, the sublingual, they are present inferior to your tongue. This is the placement of these glands. Now, what about their general structure? These all are covered by a dense connective tissue capsule, if I can mention it for you, and definitely they are well laden with the collagen fibers in it. And capsule, what it has to do? This capsule is going to give septa inside and divide the main parenchyma of your gland into lobes and lobules. And what is the purpose of septa? It has to give a passage in addition to the support, the neurovascular bundle, right? We mentioned the blood vessels, lymph vessels, nerves and various ducts are supposed to pass through it. And the parenchyma of these are basically consisting of the secretory elements. These are the acini and the duct system, right? Now we have to discuss about the general structure of the major salivary glands. These are being covered by the dense connective tissue capsule I mentioned earlier, well laden with the collagen fibers, having the septa definitely. And in the picture you are looking into the basic secretory unit of it, and that is embedded in the parenchyma. And it consists of basically the acini or alveoli, right? And these could be serous acini and could be the mucous acini, right? And the duct system you can visualize. Now we have to discuss them one by one. These acini are the you would say alveoli are rather spherical structure and secretory in nature, right? Because their cells are specialized to secrete, right? Having the central cavity. And finally they lead to a duct system, and the serous secreting acini are protein secreting in nature, right? And the mucous acini are mucus secreting in nature. And in some salivary glands we see the mixture of these two. Now we will consider the structure of serous acini, which are generally spherical. And can you consider them as a bunch of grapes? And they resemble with that of the grapes, right? And if you concentrate, the basal part of these acini are having the myoepithelial cells. As the name shows that something, a cell having the branching process is epithelial in nature, and myo means some contractile element it carries, and where it is present. If you concentrate, they are located in between the basal plasma lemma of the mucous cell and the basal lamina of this secretory cell, you would say, or basement membrane on which the cells rest, right? Now, you can see that these SNI are laden with the pyramidal shaped cells, right? And they are resting on the basal lamina, and they have got narrow apical surfaces towards the lumen. At the apex, if you concentrate, you can see secretory granules, which are zymogen granules, right? And if you concentrate, that the basal area is having spherical nucleus, and the base of your cell are well laden with the mitochondria, rough endoplasmic reticulum, free ribosomes, and they are giving the eosine or basophilic stain to these cells, right? And the ribosomes, they are also a part of it, and we can observe that the Golgi complex occupying the supranuclear position, right? And at the apex of these cells, they have got junctional complexes as well. So these apical zymogen granules, they are giving intense acidophilic stain, and the basal region is marked with the basophilic stain, right? Now concentrate on the mucus secreting SNI. They are somewhat tubular, like mulberry, right? And again, pyramidal shaped cells are a part of it, but the thing special about it, they have got the nucleus, which is flattened and oval and pressed against or near the basement membrane. Why? Because the apex of the cell is filled with the mucinogen granules, right? And they are pressing the nucleus towards the periphery. And if we give the H&E stain to it, they usually are washed out and giving an empty look to the cells. And definitely the basal part of a cell will show the mitochondria, rough endoplasmic reticulum, all the lot you can observe. Again, you can see the myoepithelial cells on their margins in between the basal lamina of the epithelium and the basal plasma lemma of your serous or mucous SNI. Now, you can see that each cell definitely are having the nucleus and organelles, and if we have to talk about the myoepithelial cells, they are showing the contractile property, right? And what they do when they contract, they push the secretion produced inside these SNI towards the duct system, and definitely have the nucleus and the organelles and the longer processes, and are laden with the actin and myosin filament that are giving contractility to it, right? But usually they are difficult to get stain, and only we see the nuclei. So this is a typical look of the myoepithelial cells. Over the, you can visualize the SNI, either serous or the mucus. Now after these SNI, you can visualize the purple color start of the duct system. The first smallest duct system is the intercalated duct. Intercalated means something inside, starting from the center of the SNI. And you can see they are lined with the simple cuboidal epithelium. Interesting fact about it, that these cells are capable of dividing and differentiating into secretory cells or ductal cells. These ducts are longer in parotid and submandibular gland, and you expect them shorter in sublingual gland. Now we have to see when these intercalated duct they merge, they form the striated duct. And still these two, the intercalated duct and the striated duct, they are also called as interlobular duct because they are still present in the lobules. So the striated duct, as you can see the transverse section of it, the striated duct are having the larger caliber. Epithelium is columnar. Simple columnar is there. You can see the deep eosinophilic coloration, and an important thing why we call them as striated duct, because the basal part of the pyramidal or this columnar cells are showing the longitudinal running striations called as basal striations, and they are arranged longitudinally having inside the elongated arranged mitochondria, and this is the reason that they are forming the striated duct. And we can see the striated duct, if you observe it under electron microscope, they could be possessing the deep infoldings of this, you would say, the basal plasma lemma. Here the bases of the cells, they are oriented in rows, right? And because they are possessing the mitochondria elongated present inside. And what are these? These features basically suggest that these cells of the striated duct are ion transporting cells, right? Because they are supposed to modify the composition of the secretion inside. How? In two ways, right? Number one, they have to absorb sodium and they have to secrete potassium and bicarbonate. And the resorption of sodium from the acinar secretion is the reason to reduce the tonicity of your saliva, right? And now we should know that the intercalated duct and these striated ducts are able to modify the secretion present inside the glands, or you can mention that they are the secretory duct system of the salivary glands. Now we have to concentrate that so many striated ducts, they are converging to form the interlobular ducts, right? And they basically lie among the connective tissue fibers well laden with the you would say the collagen fibers in them. And what they are now, basically initially lined with the simple cuboidal epithelium, and the special thing about they are just simple passage or conduits and do not now modify the secretory products. So that is why we call them as excretory ducts now. The interlobular ducts are called as excretory ducts because they are not modifying the secretory products. I am mentioning again and again. And another important fact is, now we can see that the intercalated ducts joining to form striated duct, or these two are called as intralobular. These are merging to form interlobular, right? The interlobular are merging to form interlobar, and so many interlobar will merge to form the main duct of your salivary gland, which has to open into your oral cavity. An interesting fact about it, that they are lined with unusual epithelium of various types mentioned earlier, definitely laden with the cuboidal epithelium, columnar. Sometimes they become stratified cuboidal or columnar or pseudo-stratified. What they are telling you? They are showing that these have to do some diverse function. So their atypicality would be showing that these have the cells meant for ion resorption and cells for secretion of mucus content and other proteins, and behaving as enteroendocrine cells and acting as a basal stem cell. So all diverse function present in all highly branched duct system of your salivary glands. And you know what? That the capillaries that are surrounding your secretory units, they are supposed to provide the fluid which is important for saliva production. And whenever there is autonomic nervous system stimulation and parasympathetic stimulation, what they do? They have to provoke the watery secretion, right? And the sympathetic stimulation, in addition to that, causes the inhibition of the production of saliva, and you expect the dry mouth, right? And you have very much observed it during the anxiety phase. Now we have to discuss the individual features of the glands, right? Now we are looking into the histological view of the parotid gland, which is the largest salivary gland among the three, right? And consisting of highly branched duct system. Variety of the gland, compound, tubulo-acinar, and purely serous in nature, right? It is being covered by the well-marked connective tissue capsule that sends septa inside. Let me mark these septa, and these septa carrying the blood vessel in it, and they are showing various ducts in it, right? And you can visualize these are the serous secreting acini, darker stained, and the lighter stained you can visualize these are the various ducts, either secretory or excretory. And in this parotid gland, you observe much of the adipose tissue in it. I can show it to you, which is present when the person approaches to the age of 40. And what else we expect in the parotid gland? They have got prominent intercalated ducts and the striated ducts, which are larger one, right? And its secretion is predominantly amylase, and the fact about it, it contributes 30% to your saliva, right? Now you are looking into a histological view of the submandibular salivary gland. It is addressing the mixed variety, but predominantly the serous one. Mixed means that it would definitely be showing the mucus acini as well. In the very picture, these are the serous acini, these darker stained, and if you concentrate, the lighter stained, these are the mucus acini, right? And we can expect the bunch of adipose tissue also a part of it. What else is there regarding your submandibular gland? It is again having the well developed connective tissue capsule that sends septa inside for the passage of neurovascular bundle and the ducts. And in this, if you reconsider, the 90% of the acini are serous and almost 10% are of mucus acini rather. And what else you expect? There is an interesting thing about them, that you have got the serous demillions present around the mucus acini. What are these serous demillions? These are having the half moon shape or crescent appearance. So let me show you some other good picture of the serous demillions. Yeah, you can observe that these are the serous cells, right? But if you can visualize the acini, and on its very margin there is a cap-like structure marked in a pink color. These are the serous demillions, semi or half moon shape present. It's being said that these serous demillions are present at least as an aggregation, which are be a part of artifactual problem. They say that when you give the stain, the adjacent mucus cells swell up in the routine preparation, and they are going to form the serous demillions. But some author do write that these are present as a separate entity. So we can speak about both school of thought, that serous demillions could be a artifactual fixation problem, or they may be present as a separate entity, right? Now, if you concentrate on the histological view of the smallest salivary gland, the sublingual, which is smallest I mentioned, again consisting of branched tubulo-acinar, but here predominantly the secretory tubules are mucus acini, right? And being covered by the serous demillions on its periphery. Let me show you that let me mark with an ink that these are the mucous acini, and the outer cap like darker staining are being by the serous demillions, right? And in the central vicinity hardly you would see the pure serous secreting acini, and other are various ducts. Here what you expect, the intercalated and the striated ducts are hardly you can see, but the other excretory ducts you can expect here. And the connective tissue is as a capsule is also present in a very thin or small amount, right? This is a very good representation of the salivon, or the basic secretory unit of your salivary glands. Concentrate from the top to bottom. There are the acini they have marked, and then the start of the intercalated duct. Then they have shown you the striated duct, and then the excretory ducts, right? And in the three glands, the parotid, submandibular, and sublingual, they are showing you the ratio of various cells and the ducts. The darker pinkish stain are the serous acini, right? The maximum ratio is in parotid gland, and you can see them, right? The serous acini ratio. And in the submandibular, the 90% of them, and almost 10-15% in the sublingual. And if you concentrate on the mucous acini, the number is maximum in the sublingual and 10% in the submandibular, but you hardly find any in the parotid gland, right? Then you can see the this intercalated duct I want to show you. This is of larger length, and the striated duct as well in the this parotid gland, and both of them you can expect in the submandibular, but hardly or with a very small amount you can expect them in the sublingual. And excretory duct definitely ratio is same in the three, right? That is very important diagram to memorize the difference among the three, right? This is the histological section. You can see the striated duct here, right? These are the striated duct. The basal area are showing the basal striation, and the nucleus in the striated duct is shifted centrally, because in the serous acini they were present at the periphery, near the periphery, not exactly near but a little bit towards the periphery, but in the striated duct they occupy the central position. Why? Because the basal region is there for basal striations, right? In this histological view, you can see the submandibular gland well laden with the serous acini, but there is a bunch of, I would mark, of mucous acini which is being enlarged and is shown here, right? Again, a beautiful diagram to show you the presence of striated duct. Very clear or very differentiating from that of the acini. Acini are definitely showing the pyramidal shaped cells, right? And hardly you can see the lumen. But in the ducts, you can see a clear diameter of the central lumen, right? This is a histological view of the parotid gland, in which you can see whole area is being covered by the serous acini, and you can visualize the striated duct over here, and you can observe then or identify with the help of columnar epithelium and a clear lumen in between, right? And this is the look of the sublingual gland, its histological section. You can see all the clear area well laden with the mucous acini, and their margins are definitely darker stained, showing the serous demillions. And you can observe another important thing, that the connective tissue is not very much clear in the sublingual gland, right? And secretion definitely of the sublingual gland is the amylase is there and lysozyme is there, which are adding the to your saliva, right? Now in this picture you can see that the interlobular ducts, which are secretory duct, consist of intercalated and striated. Epithelium you can mention simple cuboidal to columnar, right? And interlobular again lined with the similar epithelium, and interlobar having the pseudo-stratified columnar epithelium. Main duct can address the stratified columnar, and near the mouth or oral cavity epithelium becomes stratified squamous non-keratinized. That chart helps you to memorize.